In December 2022, we began our new BRC 5-year term, fueled by substantial funding from the NIHR. In the short film that follows, you're going to see some really magnificent examples of the outstanding science and clinical translation conducted by our BRC and our plans for the next 5 years.
My team is trying to understand how genetic mistakes can affect the function of immune cells in the body. In a group of diseases called inherited immune deficiencies, these genetic mistakes actually disturb the immune function so severely that patients have a very shortened lifespan and can die in childhood without treatment. We have developed a number of gene editing approaches that can actually correct genetic mistakes in cells and allow them to function normally in a number of immune diseases.
So what do you think are the main successes of our previous BRC funding cycle in terms of advanced therapies for neurological diseases?
I think one of the most exciting things, certainly now and for the future, are these gene therapies based upon viral vectors that are being used across all neurological disease. We've shown preclinically, for example, that we can alter the excitability of the brain. And what was exciting was that it was possible to show that you could in fact cure focal epilepsy, which starts in one part of the brain, in these preclinical models. These trials really are revolutionising the way that we view neurological disease and the way that we can treat it.
We'll be embarking on the Octopus trial, which is funded by the UK MS Society, a multi-arm, multi-stage trial.
That's very important, no? Because you can do faster trials with more drugs, so you can find the treatment sooner.
Because phase 2 plus phase 3 take a long time, could take 10 to 15 years, and this has the opportunity to maybe halve that.
For a number of dementias, we know they are caused by a gene, and we now have ways of reducing that devastating pathogenic effect of that gene mutation. These gene therapies can attack the proteins that we've known for over 100 years to cause diseases like Alzheimer's disease. The hope is there's a benefit in that vast majority of cases that are not genetic, which is really exciting.
The CAR T cell that we've developed for leukemia, it's a particularly novel type of CAR T, so patients tolerate it well and it's very effective. It can protect the patient from relapse from their leukemia months and years after this one infusion of CAR T cells. So for us, that's a huge achievement.
The funding that the BRC has contributed to essentially establish and maintain the CAR T cell program has been one of the reasons why CAR T cell therapy is so prominent in the UK.
As a consequence, we've been able to open more clinical studies than anywhere else in Europe, and we've got patients now who are alive for years following their therapy, disease-free. Being able to create a spin-out company enables UCL to roll the science out to a bigger patient population. That's hugely important to the future of the science that's developed at the Cancer Institute.
The big aim of our program is to move from blood cancers into solid cancers.
We've got a whole range of different CAR T-cell therapies that we're proposing for solid cancers that we're developing in the lab at the moment.
In the last 5 years, there's been a transformation in the way that prostate cancer has been managed, and a lot of that has been led through UCL and BRC work. The BRC have supported the delivery of the Verdict MRI study. So this is a technique which you can use together with multiparametric MRI. And we've shown in studies we could potentially avoid 90% of unnecessary biopsies.
About 1 billion people globally have high blood pressure, and for many of them we don't know the underlying cause. However, for maybe 10%, there is an underlying cause that is potentially treatable and curable. The problem is, it's very difficult to diagnose. We have developed our own PET tracer which highly selectively binds to aldosterone and identifies this tumour. This is something that we can't normally see, and here you go, that's pretty difficult to miss. And this patient's now going to go to surgery, have that tumour removed, and we're very confident that that will cure this patient's hypertension. Now that is going to completely transform the diagnosis of hypertension, and this test will be available for the first time in the world at UCLH.
A specific area that was relevant a couple of years ago was vaccine-induced thrombocytopenia and thrombosis, which was linked to primarily the first dose of the AstraZeneca vaccine. We were the first to identify this condition in the UK along with colleagues in Birmingham, and also set the standards for treatment, altered policy, and within 6 months we had no further cases of this life-threatening condition.
People with sickle cell and thalassemia are very dependent on blood transfusion, and a particular side effect they have is the formation of antibodies. That can affect the ability for us to transfuse you, and it can lead to increased risk of death and illness. So what we want to do is improve the delivery of transfusion using artificial intelligence and genomics to deliver better matched bloods for our patients.
The current focus of our research has been on which diseases co-occur in individuals and why. We link health data recorded in the NHS to research data like UK Biobank. This is providing the opportunity to really understand the molecular causes of disease, and through that common pathways and common treatment targets.
I think one of the key things that the BRC does is bringing together doctors, public health specialists with people who understand data, and it's really integrating those different kind of skill sets. One project that we've been running in the BRC is applying AI to data from the hospital to try and tackle the problem of antibiotic resistance. The aim of Samurai was to try and find people who didn't need to be on antibiotics using machine learning. And so the algorithm finds the people, predicts which people might benefit from stopping, and then flags them up in the system so a clinician can go and review the patient and stop antibiotics early.
Tuberculosis has been with us for thousands of years, but it's still amongst the most common infectious diseases to afflict people worldwide. But importantly, most of the people who become infected don't go on to develop disease. And the really big challenge is to work out which of those people who become infected really benefit from antibiotic therapy.
We know that preventative antibiotics themselves usually have to be taken for between 3 and 6 months. And we need to be quite mindful of the burden that that puts on patients and the risk of side effects. So Periscope TB essentially takes that test result, accounts for other patient factors, and then generates personalised estimates of somebody's future risk of TB both with and without preventative antibiotic treatment.
I think what's unique about the BRC is that we can look at something which is a little bit out there. It may or may not work, and we can take a chance and we can put together a really exciting, innovative, cutting-edge program and resource it and see whether we can really make magical things happen. And as you can see, more often than not, we succeed.
It's the kind of thing that you hope to have the opportunity to do as an academic, but very rarely happens. It's so unusual to be able to do research and see it translated into impact, especially that quickly.
There are neurologists and immunologists, respiratory physicians, cardiologists, data science people, and we learn so much from each other.
We do have a new scan which we've developed. It's a 5-minute scan of the prostate. What if we could deploy this within the general population as screening for prostate cancer? We can maybe capture those men who would otherwise never get in to see us until it was too late for them.
We have patients who have relapsed refractory disease, so disease that has failed standard treatment, and a significant proportion of these patients can now be cured when they wouldn't have been cured in the past.
It does feel like a momentous moment. I have spent 30 years particularly studying families with these dementias, and for the first time I think we can really say to people, yes, those therapies are now within reach, they will be available for you, and at a personal level, that feels amazing.